All right, so this is an inaugural address. So the Don Brooks and Dana Devine Transfusion Science Innovation Address, and Dr. Steve Withers will be giving this address. It's really very appropriate. Steve is an emeritus professor of chemistry and biochemistry at UBC, where he was on faculty for 40 years. And he's still on faculty because he's bicycling around. You'll see him often like that. He's still got a group of young scientists with whom he is working. He's collaborating widely. So he's certainly not emeritus in the way some of us think emeritus is. He received his Bachelor of Science and PhD in chemistry at the University of Bristol in the UK. And then was a postdoc fellow in biochemistry at the University of Alberta. I wasn't there, but he was recruited to UBC subsequently, where he rose through the ranks, as you imagine, very quickly. He has held numerous leadership positions, in particular driving the creation of UBC's Genome Science and Technology graduate program and the Center for High Throughput Biology. His research really is perfect for this sort of award, where it sort of bridges basic biochemistry and chemistry with applications that can be clinically relevant, and in this case into the blood transfusion world. He's an expert in, I call sugar chemistry, but it's carbohydrate chemistry and enzymes, and creating enzymes, all kinds of sugar cutting enzymes that can be adapted and modified, and I can't tell you anything about it because I have no clue on how to imagine what he does, but it's a puzzle. But we're really pleased that he can give this address. So Steve?
Great. Thank you very much, Ed, for that kind introduction, and thank you for this great honor to receive this, or to give the Don Brooks and Dana Devine Innovation Address. I don't know Don and Dana as well as many of you people do, and Cedric's given you a wonderful overview of their lives and careers, but I have known both of them for some time. Don was actually or is actually a colleague of mine in chemistry. He has a 17% appointment in chemistry, yet for that small percentage appointment, Don delivered way more than 70% of his time. In fact, you know, Don's a big guy, so 17% of Don is a lot anyway. So I would meet Don mostly in committee meetings, thesis committees, etc. And Don was always the sensible guy, the guy who would give the sage advice and make sure things didn't go off the rails. So greatly appreciated that. Dana, I didn't really get to know Dana until my collaboration with Jay, which has been a fantastic collaboration, sort of got me into the field of blood. And Dana was a wonderful help in our project. She's connecting us with people, dealing with the press when they got interested in the work and things like this. So huge respect for both of these people, and I'm delighted to be asked to present this address, excuse me. So what I'm going to tell you about today is work that I've been doing with Jay, where we're working on trying to develop what one might call universal blood, and in fact universal organs, through the use of enzymes, which are not really scary yet. It's okay. It's good. Okay. So now I feel kind of awkward speaking to you lot. I'm a chemist, right? What do I know about blood? You guys know way more about most of this. So when I screw up, please forgive me. But let me just see if I can get oh, that'll do. Can we see that? Yeah. So our blood types are determined by sugars. Sugars are super important. These sugar structures are present on red blood cells, but also on all our tissues and organs. And specifically what I'm going to be talking about today are the sugars that define the A, the B, and the H antigens of our blood types. You probably all know these structures well, but if you're of A blood type, you have this structure on the surface of your red blood cells, approximately 1 million copies on each red blood cell. And what distinguishes A is this terminal sugar here is, whoops, I should be careful doing that. That terminal sugar there is what we call GalNAc, N-acetylgalactosamine. Whereas if you're B type, you have a galactose at that position, and the only difference between the two is an OH group at this position versus an acetamide group, but yet we know the physiological consequences of mixing those two up. If you're O blood type, of course, you're just missing that terminal sugar, and you just have the underlying structure here, which is the H antigen. Okay, as we all know in this audience here, type O is universal donor blood, so the idea has been out there for quite some time that if one could find a suitable way to clip this terminal sugar off the A antigen in the context of red blood cells, you should be able to turn A red blood cells into O-type red blood cells, which are universal donor red blood cells. Similarly, if you could find a suitable enzyme to chop off the galactose from B, you could convert B-type red blood cells to O-type red blood cells. And our assumption is that that's the only difference between A, B, and O, is that terminal sugar. Now, as I said, this idea has been out there for some time. Goldstein in '82 was the first one to demonstrate this, using some enzymes, or the only commercially available alpha-galactosidase, which is the enzyme that would be needed at that time, which was actually from a coffee bean, strangely enough. Far from ideal enzyme, but by throwing enough of this enzyme into the red blood cells, he could convert enough of the B-type red blood cells to O to do a small clinical trial and show that this was successful. No significant bad things happened. At that time, there were no commercially available N-acetylgalactosaminidases. That would be the enzyme you need for this. So fast forward to 2007, and a wonderful paper came out from Henrik Clausen's group, performed in conjunction with a company called ZymeQuest out of Boston, where they screened a library of bacteria looking for these enzyme activities and were successful. They found enzymes that would cleave the A antigen, or cleave the sugar off the A antigen, and separately other enzymes that were better at cleaving the B antigen. And they took this forward a fair way, but still these enzymes were not quite active or perhaps specific enough to go into practical use. So this is where we thought we might be able to play a role. We wanted to see if, using our knowledge of enzymes and screening technologies, which I'll introduce in a second, we could find better enzymes of significantly higher activity and specificity that might get us over that barrier to being good enough for the job. We wanted them to be highly active, highly specific, work well at pH 7, and ideally work in whole blood, which the existing enzymes did not do. Okay? And that will be useful for some of the applications. So the approach that we've used in this is an approach that we've learned from our colleague Steve Hallam in the microbiology department at UBC, and it's the approach of metagenomics here. Now, traditionally, if you go searching for new enzymes from bacteria, you would do what the ZymeQuest group did, which is you get an array of different bacteria, each of which is grown up independently, so you have to be able to culture them, and then you would screen that array of bacteria looking for the activity of interest. Now that's all fine and dandy, but only a tiny percentage of the bacteria that exist around us can actually, or have actually been successfully cultured in that way. If you could look at all the bacteria, all the genomic content of all the bacteria, then you would find way more enzymes. They may not work, but you should have a better chance of finding good enzymes. And this technique of metagenomics, which Steve was an expert in, or is an expert in, is what we have applied to this approach here. So in this approach, what you do is you try to think of what environment, it may be it soil or ocean or feces or whatever it might be, what environment is most likely to harbor bacteria which themselves would be harboring enzymes or producing enzymes which would carry out the sort of reaction you want. Okay? So you think about that first of all, and then you go to that environment, collect some of the sample, And now, instead of trying to culture up the bacteria within there, what we're going to do is just lyse all the bacteria within that sample and pull all the DNA out. Okay? I say all the DNA, it'll be a minuscule amount, but lots of information. Now, we don't care about which bit of DNA came from which enzyme, because I just want the genes, right? I just want the enzymes in the end. Ultimately, we can probably find out where they came from, but that's not the primary interest. So we take that DNA, and now what we're going to do is chop it into bits, either small inserts, as we call them, which are a bit larger than an average gene, and then put that in on plasmids into an E. coli library, or we use larger inserts. We more carefully degrade it to make large insert libraries which contain multiple genes. And in the work I'll tell you about here, we actually use these large insert libraries, which have been very, very good to us. Now, what we're doing is we transform E. coli with these bits of DNA. And now what we're doing, of course, is crossing our fingers that E. coli is going to read those genes, or at least some of those genes, and produce the protein products, and specifically the enzyme products that we're interested in, or that we're hoping to find. So now it becomes a screening approach, and I got very involved in high-throughput screening from my involvement with the Center for High-Throughput Biology. I believe that was sort of the future in many ways of enzymology, and it has certainly proved to be good for us. Now, in our first approaches to this, and actually the approach that we use in the work I'll tell you about, what we would do would be to take a colony picker, pick those individual colonies into 384-well plates, and in the particular approach story I'm going to tell you today, we created 20,000 clones in this fashion, which is something like 50 384-well plates, using our colony picker. That approach is fine, and it works actually very well, but once you've got 20,000 384-well plates, that constitutes about 2 liters of solution that you need to fill each time. Now, the 2 liters of buffer is not an issue, but when you need 2 liters of very expensive substrates, that becomes impossible, basically. And you'll see that some of the substrates we use are quite complex. So what we've been moving towards, and we're just starting to get it to work now, in fact, just got some great results yesterday. My student was using less than 1 milligram of a substrate, screened a 20,000 library, okay, using this approach. So what we're doing is using microfluidic droplets as tiny, tiny reaction vessels, okay? Using these microfluidic droplets, which I'll explain a bit more, we can reduce our substrate needs about 1,000-fold. So now with less than 1 milligram, we can screen 20,000 clones. So what does that involve? I just throw this in because you've had lots of blood stuff. Let me tell you some other science. Okay, so these microfluidic droplets are made by a very simple microfluidic device, which looks kind of complex here, but really it's basically a 4-way junction. You have an aqueous flow stream, which is going to contain your bacteria, okay, along with any substrates you need to assay. And then you have an orthogonal pair of oil streams coming in, and you flow these two at just the right rates, such that as the oil flows in and as the water flows across, it pinches out little water droplets within an oil layer. And here you see these little water droplets, incredibly regular droplets. Now, each of these little droplets is a little reaction vessel, a little screening vessel. Now, what you need to then do is adjust the concentration of the E. coli such that statistically you're going to have either 0 or 1 E. coli per droplet. It's not that hard to do, in fact, at the end. 0 or 1 per droplet. And then, if you do it right, these droplets are really quite stable. We keep them for 1, 2, 3 days even. You just have to aerate them a little bit. You let the E. coli actually grow within there, produce their enzyme, and then, as I said, you have incorporated the substrate in there, so you look for fluorescent colonies forming in that way. So this would give an example of what you'd see in a bright field. These would be the droplets, but now look at them under a fluorescent light. All these little fluorescent dots, which you see are very real discrete dots, are droplets that contain the functional enzymes. So then the task is to pick those droplets and then just grow up the bacteria and sequence, okay? Sounds simple, it's actually quite tricky, but I think it will work. Okay, now when we first did this, Peter, my postdoc, who developed this in our lab, others had developed the technology elsewhere. Again, we're using the technology but adapting it to our case. He just wasn't allowed coffee for it that morning. And then he would go in with a pipetter trying to pull out these individual droplets. But he finally rebelled and said this was too hard, especially hard on the eyes. So we've now, and this is largely through the efforts of Steve Hallam, UBC now has a droplet sorter which works like a fax sorter, but it works in an oil medium. If you take an oil medium along to the fax sorting people, they would not be very pleased with you. So don't try that at home. If you need to do this sort of sorting, go and see Steve. And these droplet sorters do basically just like a fax sorter, okay? All right, that was a little digression. So where are we going to look? What environment are we going to go to find enzymes that might cleave the A antigen? And we're focusing on the A antigen because that's 40% of people. Well, it's going to give us the biggest bang for our buck if we can convert A to O. We actually look for A and B, but particularly A. So the environment we chose to look in was the human gut, because, as I mentioned, this A, B, and O antigens are expressed on all our organ surfaces, including the gut wall, in the mucins of the gut wall, along with many other antigens there. And so bacteria in our gut, which are known to degrade or to live upon the glycans that are being produced, especially at times of sort of starvation of food for them from eating sources, that those bacteria, some of those bacteria at least, must encode enzymes which are doing this degradation. So if they can cleave the GalNAc off of mucin, maybe they'll be able to do so off red blood cells. That's another big question mark. So into the workflow of the approach. So Peter went and found a donor, in this case not a blood donor, but a poop donor, and he chose somebody of type AB blood, just hedging our bets about correlations between blood type and microbiome. AB donor made a library of 20,000 in the way I briefly described, and then what he elected to do was to screen using very simple monosaccharide substrates, which just represent the sugar at the terminus there. Ideally, we would use a big oligosaccharide, but we can't make enough oligosaccharide to do that. So we use these simple monosaccharide substrates, just hoping that our enzyme will be active enough to cleave these, and we mix both the GAL and the GalNAc in at the same time, and screened, and Peter got 226 hits out of the 20,000, okay? That's why you need big numbers, okay, initial screen to find the things you want. He then made a hit plate, which is actually what this data is, and deconvoluted by then rescreening this hit plate just with the individual substrates and identified who was GAL and who was GalNAc cleaving. Now we'll see which of those, if any, can cleave in the context of the A or B antigens. And so now we come in with oligosaccharide substrates, which we synthesize in the lab. We synthesize them enzymatically now. But we can make in the 5, 10, 20 mg range. It's hard to make much more than that, but that's plenty to screen a few plates. To actually assay these, since our enzyme is going to just cut off that terminal sugar, that wouldn't cause it to become fluorescent. So what we use is a coupled enzyme assay, classic enzymology here, where we throw in the enzymes that will degrade, no good me pointing at the screen, is there? Which will degrade the oligosaccharide that's released once this one has been cleaved. So we throw in the enzymes that will cleave all these bonds down and release this fluorophore, okay? So that's a classic coupled assay, and it works beautifully, and we can do all that in the droplets. Everything is stable, works fine. And in that way, we were able to identify 7 hits for B, 8 hits for A, and 1 that cleave both. Now, I know it's going to be short on time, but I'll just say that the B hits all turned out to be false positives. That's an interesting story in itself. But the A hits were real, okay? So let's see what we do. So the next step in this is you need to sequence that insert of DNA to see what you've got and do a bit of bioinformatics upon that. So we sequence the A cleavers, as I call it, and what I've just shown here is we've highlighted in color anything that looks at all like some other glycosidase that's been identified before, okay? And what we see is that The majority of them are yellow, and you'll see that, in fact, these are really the same gene in different contexts. This is another similar gene in another context. These yellow ones are in the same family as the enzyme, the best enzyme that was discovered in that 2007 paper, okay? Same family as that. So Peter, first of all, cloned out representatives of each of these, checked them out, and they were good galactosaminidases that worked on the A antigen, but they didn't work on red blood cells. These were really mucin-degrading enzymes, okay? So that was a bit of a bust. Whoops. Fortunately, there's two other hits, otherwise I wouldn't be talking about it. One of them here with this big blue arrow, which actually belongs to a sequence-related family which is known to contain galactosaminidases. We checked that one out, and it did cleave the A antigen on red blood cells, but it was pretty slow. It was no better than the best one that had been reported. So it's just left us with one, but we only need one. And this one had 3 different genes in it that were related to things that do things to sugars. So Peter cloned each of those out, tested them, and none of them worked, which was a bit of a shock. But he's a smart dude, so he started mixing and matching, and quickly found that it required 2 of those enzymes working together to cleave the single sugar off. And again, to cut a long story short, what's actually happening, and here's a little bit of chemistry for you. This is the A antigen again with the GalNAc terminus. That first enzyme that works is a deacetylase that cuts off the acetyl moiety off of the GalNAc, liberating galactosamine. Okay? The second enzyme is a galactosaminidase, which cuts this galactosamine moiety off, liberating the H antigen. So it's a 2-step process, and you might wonder, why is he excited if the other guys can do it with one enzyme? But these enzymes are super active, okay? And that's what we wanted, was high activity. You're a blood community, so something I became aware of is sorry, I'm blanking on the name. I'll come back to that later, sorry. Just lost my focus there a second. So these enzymes look interesting, and we know they will work in the context of red blood cells. I've thrown myself off. So what we need to know is not only are they active, but are they specific? So we started out with the deacetylase. This is now purified enzyme. What we did was screen this against a whole range of substrates that contain a terminal GalNAc. And what we find is that the GalNAc is only cleaved if the substrate is an A antigen substrate. So anything else that contains GalNAc, it doesn't touch. So that's wonderful news. We want to not be hitting anything but A antigens. Okay, and all the different subtypes of A antigen are cleaved too. We wanted to understand the structural basis for this, so in collaboration with Natalie's group, solve the crystal structure of this deacetylase, and we see indeed it's a classic metallodeacetylase, and we can understand the specificity for the A antigen because of the specific recognitions with the fucose moiety, which is what really defines the A antigen there. So we can understand that. The second enzyme, the galactosaminidase, we did similar specificity studies on galactosamine-terminated substrates, which we generate by just deacetylating the previous substrates. And we find that this enzyme has relatively broad specificity. But that's okay, because there'll only be galactosamine if the first gatekeeper enzyme has done its job, and we know it's specific. So that's good news. Okay, so this all works well, and the fact, that's kind of partly takes me back to what I was going to say here. Galactosamine looks a lot like galactose, right? That NH2 looks much more like OH than does the acetamide, and that's why this enzyme has low galactosidase B cleaving activity too. That's what I also wanted to say. Okay, I'm going to come back to that later. So the galactosaminidase has broader specificity. Again, in collaboration with Natalie's group, we've very recently solved the structure of the galactosaminidase. We, in fact, hope to submit this weekend, if possible, the paper. What's kind of cool here, and and this one is for Chris overall. There's 3 different metals in the protein, okay? There's a zinc and a manganese, which seem to be structural, and there's a cobalt right in the active site. The only cobalt-containing glycosidase I've ever heard of or ever find anything about. And that cobalt is involved in coordinating the amine of the substrate. It actually helps build that specific galactosamine binding site by the amine of the substrate coordinating directly onto the cobalt. Anyway, that's kind of cool, a little enzymology geekiness here. What you want to hear about is do they work on red blood cells? And how well do they work? So we're using the MTR cards and FACS analysis, and this is all work all done in Jay's lab, to test this out. And what we found was that the previous best enzyme, which was the 2007 paper top candidate, works under our conditions here if you chuck in 15 micrograms per mil of enzyme, but only does so in the context of a macromolecular crowder, such as dextran, okay? So macromolecular crowder, which helps to push the enzyme onto the cell surface, allows us to cleave with about 15 micrograms per mil of the previous enzyme. Our enzymes, a pair of enzymes, work at 0.5 microgram per mil each, okay? So depending on how you want to count that, that's 15 to 30-fold better than the previous best enzyme. In fact, they work even without the crowder. You just have to add a little bit more. They generate the H antigen as expected. They work in whole blood, which is super important. And we can remove the enzymes by centrifugal washing of the red blood cells. So these work on full units. A few milligrams will convert a full unit with no problem. And Jay's lab's been working with actually a lot of help from Dana on this initially, on using the hemanetic cell washer as a way of removing the enzyme from the red blood cells. And that seems to be working well. Jay's lab's been sort of optimizing that process. Jay's also been involved in a lot of cross-matching studies, and I feel really embarrassed to be talking about this here because I know very little about this part, but suffice it to say, they've done a pile of cross-matching. There is a little bit of a cross-matching issue when they're mixed at 1-to-1 ratio, so not when they're at much higher dilutions. This same sort of phenomenon of a cross-match against O serum had been seen by Goldstein and by the ZymeQuest group. But they did successful clinical trials. That didn't seem to be clinically an issue. So again, Dana, we had a lot of conversations with her about this, and she suggested that Jay's lab get the monocyte monolayer assay going, which I'm even less confident talking about here, which apparently is a much more clinically relevant assay. And these that sort of are a bit shaky on the cross-match studies pass this test fine. So it looks like we're good in that regard, but it's something that we continue to work upon. What is the basis for that? So that's the A. We've just very recently discovered some B-cleaving enzymes that work through a combination of droplet screening and bioinformatics approach. And so that can move forward and allow us to have both enzyme activities, the ability to convert both A and B. We're working with the US Army, actually the US Air Force strictly. It's funding us to try to get this to work on the battlefield, so to speak. Again, you guys are the experts for that. My understanding is that in cases of severe traumatic injury, patients do better if they're given whole blood rather than red blood cells. So they want to know, can our approach work in whole blood? We know our enzymes work really well in whole blood, so that part of it is fine, but we have to find a way of getting the enzyme out of this whole blood. We can't just do the centrifugal approach. So what Jay's working on is developing filters which could be put in line and such that when you could imagine, I love these little cartoons, the blood being taken from the walking blood bank here of somebody, say, of A type, converted in the bag with enzymes that are sitting in the bag, and then prior to administration, just run through the filter into a separate bag, and the patient's treated, and all is better. I wouldn't say well. Okay, so that's where we're going with that. Am I okay for time? Good, okay. So I mentioned earlier that these same antigens are present on the surface of all our organs. So the question crossed our mind while we were putting the patent together, could this possibly be used for organ transplantation too? Because when matches have to be done for matching a donor organ to a recipient, the ABO blood type, of course, is, as you all know, is the primary match that has to be made. Obviously, the HLA antigens have to be matched well too, but the ABO is super important. And it's this combination of matches that makes it difficult to match a donor with a recipient, which results in very long waiting times for patients. Kidney transplants are the most common transplants done. But if you need a kidney transplant, you could be waiting from 2 to sort of 5 or 6 years to get an organ. Sadly, during that time, a lot of people are not going to make it. They're going to die before they get a donor, get an organ. Notice that the amount of time you'll wait differs very much depending on your blood type. Okay? So there's a lot of sort of asymmetry and essentially unfairness here. Why might that be? Well, donor O-type kidneys can be given to anybody. Okay? So those can be used across the whole lot. So if you're an O-type recipient, you're less likely to get to receive a kidney because of that fact that your kidney donor pool is being shared out to some extent across. So if there's a way that this could be, that our approach could be used to help that situation, that would be a wonderful contribution. Now, when we'd first thought of this, I thought this was a waste of time. Because by contrast with red blood cells, which will not regenerate the antigen, the organ will regenerate that antigen. On the time scale, we don't know exactly. On what time scale, we don't know exactly, but it would regenerate that. So I thought there would be no point to this. But fortunately, I got talking to some transplant surgeons, and they said, hold your horses here. It's not as simple as that. This might be really useful because, as I understand it, their main problem is in that first hour or so after transplant. If you transplant an organ in that has an incompatible antigen on the surface, then antibodies in the recipient's bloodstream that recognize that will glom onto the surface of that organ, and as I understand it, within a matter of hours, that organ will be turning black and rejected, okay? They said, if you can get us through that first hour or so by stripping the antigens off, we can probably control through immunosuppression the antigen, or the immune responses as that antigen regenerates. And then hopefully through the process of accommodation, which can occur whereby foreign antigens are gradually tolerated, especially antigens of the carbohydrate type are well or increasingly tolerated through this process of accommodation, maybe this would all work out well. And the basis for being moderately confident on that is that, particularly in the case of kidneys, there are quite a few transplants that are done across the ABO barrier. If the donor kidney happens to have a low titer of antibody, and if the recipient undergoes plasmapheresis to lower their antibody levels and potentially some other processes. So it's a complex process, but it is done, and the success rates on that almost mirror that of compatible transplants, okay? So accommodations occurring there, perhaps it can occur across the full range if, in cases with higher titer and without the antibody removal process, which apparently is not trivial. So the first question is, how do we do this? How can we get the antigens? How can we get our enzyme into contact with those antigens? Well, this is a case, again, of technologies coming together somewhat at the same time. Organ transplantation, or in organ transplantation, there's increasing use of these perfusion devices to keep these organs alive and going, or in better shape, for longer time periods between the donation and the transplant time. In fact, during that time, they even kind of fix up and tune up the organs a little bit with maybe a touch of antibiotics or something to clear some potential blood clots and such like. So this, through conversations with Marcelo Cepal, who is our lung transplant surgeon in Toronto, He was the one that really turned me on to the idea we should be able to try this. He said, if we chuck in the enzyme into this perfusion fluid while it's going around our lungs, maybe that will clear, and that's a good opportunity. So we wrote a CHRP grant, successful in getting funded, and went ahead with this process, and it works really nicely. If he chucks in as little as 1 microgram per mil of enzymes into the perfusion fluid, and he's done this on, last count was it 10 or 11 different donated human lungs. And I should be really clear, these ones were lungs that had been donated for transplant but deemed not viable for other reasons for transplant, okay? What he showed was that these, as I said, these lungs cleared almost entirely the A antigen off of that surface. We see 98% clearance of the A antigen from the surface of the lung. So it's feasible to remove that from there. Is there going to be any benefit? Clearly, we have not been able to, or he has not had approval to put any of these lungs into a recipient yet. But he's done a sort of a model of what, to model to some extent what might be seen in those cases. So he took a pair, did this in several instances, a pair of lungs of type A, split the lungs, have one as control and one as treatment. Whoops. The control lung receives no enzyme, it's just perfused. The other one is treated with the enzyme. And then what he's going to do as the model is to float through that lung A-type blood, or actually strictly A-type plasma, and see what happens. And the first indicator is that the anti-A antibody in the control just gloms onto the tissue and depletes from the plasma, as you would expect, okay? So that occurs, whereas in the enzyme-treated lung, there's no change in the level of the antibody in the plasma. So that just basically shows that everything is working the way it should. He sees differences in inflammatory cytokines in the two cases, and he sees better physiology in the treated system versus the control, airway pressures and things I didn't fully understand. And he sees somewhat lower edema, but the edema is pretty awful anyway because you've ripped this thing out of somebody and put it in there. So it does look promising. It looks like the approach can strip these out. It works in kidneys too. We've collaborated with Dr. Michael Nicholson at University of Cambridge, showing this for kidneys. He just published this just last month in Nature Communications. It's really a very, very similar set of experiments to what I just showed on the lungs. Something I forgot to do at the beginning was make a conflict of interest disclaimer. We formed a company, Jay and I and a few others have formed a company, Avivo Biomedical, to develop this technology. And Avivo has been the ones that are now providing this sort of a clean enzyme for these approaches. Okay, so that's my story. We're looking for permission to move to the next phase in sort of subclinical or clinical experiments on this, on the transplant system. We're working solidly with the US Army on testing out our approach on whole blood, etc. All that remains for me now is just to thank the people involved, primarily Jay, who's been a fantastic collaborator through this time, my transplant collaborators, Marcelo, Michael Nicholson, and Laurie West at the University of Alberta, whose work I've not mentioned, but Laurie was part of the team that did the first ABO-incompatible transplants on infants who have poorly developed immune systems, and was able to show that that could be done. Thank all the people in my group, but particularly Peter Raffeld, who was the person who drove this whole project and is the Chief Scientific Officer for the company, a range of other people in my lab. People in Jay's lab, particularly Hazel, who's been doing a lot of the blood work. Aizhu, who was key in Marcelo's lab. Natalie and Liam for their work on the crystallography, and these various funding agencies for their support, and you for your attention.
Transfusion. Stephen, you want to start? And then, let's see. Oh, all right, Haley, go ahead.
Thank you so much. As someone who gives a lot of transfusions, often it's not the ABO group that we worry about. It's minor red cell antigens, some of which are carbohydrates, some of which are proteins. Do you envision this as an approach where we could target specific antigens with a cocktail of enzymes, or should we be thinking upstream, looking at limiting antigen expression to make transfusions more universal for our patients?
Yeah, so just to clarify, I think my hearing's not that good, so I think I'm a little different from hearing all that, but our approach is, yeah, I should say here, sorry. Our approach, of course, only removes the ABO. Protein antigens like rhesus, we can't do anything about with this. We have some other ideas we're working on that may deal with that. But at this stage, what starts as plus ends as plus. You know, A plus goes to O plus, etc. I doubt this would be developed for any even other carbohydrate antigens just because of the expense of taking something through all of that process. So I think some other approach is likely to be the better way to go there.
Very great talk, Dr. Withers. I have a question about the kidney transplant. So, like, your enzyme only clears the carbohydrate alloantigen, but what about the protein alloantigens? Would they have an effect on the
Are you saying the protein antigens?
Yeah.
Yeah, we don't do anything about that. That's just what I was just saying there. So what starts as rhesus positive, for example, ends up as rhesus positive. But other technologies, I think, have the opportunity other technologies which might work well with low-lying antigens would have a better way of doing that.
Thank you, great work. One quick question about your method. When you were doing the droplets, so there's oil around it. How did you overcome the problem with the hydrophobic, oil around, and then you want to add the sugar, which is, hydrophilic?
Sorry, I'm not quite catching the question.
The solubility of the substrate. Around the oil around it.
Yeah, no, you bring up an interesting point, actually. So really the question comes down to, we have a water droplet in an oil layer. What if our substrates are soluble in the oil? And that is a potential huge problem you have to worry about. Fortunately, our substrates all have sugars on them, which don't like going into oil. But in our first attempts at this, we thought we were plain sailing because our substrates stay in the water droplet. But when the enzyme cuts the fluorophore off, the fluorophore is very oil soluble and just disappeared out. So we're chemists, fortunately, so we went back and redesigned our fluorophores to put charges on the fluorophores, and then they stay within the droplet. So exactly. No, it's a very perceptive question.
All right,
Thank
thank you.
you, Dr. Withers, for a wonderful talk.
Sure.
I'm wondering about the transfusion.
Oh, there you are, right. Sorry.
So I'm wondering about the transfusion part, like when you're stripping an antigen and that eventually will accept the organ, but what will happen eventually later when that antigen will express again? Is there any later chance of rejection of the organ?
So that was what had worried me about even thinking about the approach from the beginning. But this is where our transplant surgeons have to do their job. They use immunosuppressives to control that. And then this process, a process of accommodation, immune accommodation, which don't ask me about because I tried to read about it and understand it. And it seems even the immunologists aren't really clear on what's going on. That process may solve the problem for us. But it's an experiment that's waiting to be done. Obviously, as this audience knows, we're handicapped a bit by the fact that other animals don't have the ABO system, so you don't have anything other than higher primates that you could go in to test this in, unfortunately. So that's where we're working with the FDA and Health Canada and people to try to see how we can go forward.
Terrific, Steve. I know you're not a transplant surgeon. So you got
Definitely not.
You've got a good caveat there. And you've partially addressed it there and also in your talk. I would have thought something like a lung would have been an easier task to get the stripping off, because you've got tennis court size of surface to strip off and the vessels. Something more solid like a kidney would be a tough one to start with, because as I understand it, and I hope maybe a transplant surgeon's in here, that the reperfusion, when you get the reperfusion, is not just the endothelial cells from the blood vessels. They're leaking, and you've got everything getting into the interstices of the tissue, and you get a lot of tissue there to strip deep down. So the deeper tissue past the blood vessel, is that not an issue, or is it just a so small amount in that 1 hour it's not going to cause the blackening?
You are right that is more of a concern, and that's in part why we started with lungs.
Yeah.
Mostly we started with lungs because Marcelo got in touch with us first, and because actually perfusion devices were better developed and first developed for lungs, because I think the oxygenation issue isn't an issue in that case. My view is that the enzymes that are clipping this off are smaller than the antibodies that would be recognizing them in an immune response. So I don't know that there's actually a problem.
No, I'm not worried about the enzymes. I'm worried about the ones they can't access deep into the tissue, that when you reperfuse in the patient's blood's going through and the fluids are going through, then and everything's a bit in a shaky state, and so the plasma is going to get into, and the cells might get into the deeper tissue, deep within
But what is it that's going to be interacting with these A antigens that's going to be causing the issue?
Well, the leukocytes of the patient. I mean,
Right. So don't you think our enzymes are quite a lot smaller than those leukocytes?
Yeah, sure. But can you get through the whole tissue? Can you clear all the deep in?
I guess that's what we're going to see.
Yeah.
Could I interject? I'm not a transplant surgeon, but I've been involved in cases involving plasma exchange where there's been an ABO mismatch. And I think the issue is that it's not the organ tissue that has the A antigen, it's the endothelium. And the source of that A antigen is from the blood. It's not from the tissue organ itself. So if you take that organ out of the donor and put it in an O recipient, you just have to get them through the 2 weeks it takes for the endothelium to turn over for that A antigen to be just gone. So that's my understanding, because I think when they've done ABO mismatched transplants, like you said, they just apheresis the patient for 2 weeks to remove the A and B antibodies, and then after that, you can just leave it, and the organ's fine. So I think you're just doing the opposite. You're taking the A antigen off the endothelium of the recipient, or the donated organ, but you don't have to do anything beyond that.
Thank you.
Yeah.
Thank you.